Hello, everyone. Hope to find you in healthy state of mind. I should rather say. I was trying to record this video since so long, but I couldn't do it. But today I am finally doing it for you guys. And in this video, we will be discussing how important it is for us to know where how this RNA is integrating into the human genome. Earlier, it was a debate whether it is getting integrated into human genome or not. But now it is not a debate. It has been proven by the scientists, and the research paper is out also. Couple days back when they have showed how the this viral genome is being integrated into the human genome. We all know that this sudden emergence of this severe respiratory disease caused by this novel coronavirus becomes a public health emergency, right? It is a single stranded enveloped virus which belongs to the family of coronavirus family, and this is more pathogenic form. This is as compared to the ones we have in the past, the SARS we had the MERS, right? All three, the SARS, MERS, and the SARS-CoV2, they all belong to the same family, but the SARS-CoV2, the one we are facing now, is the most pathogenic form, and that has taken face of the pandemic, not just the epidemic but the pandemic, right? And the SARS-CoV-2 virus shares a high degree of genomic similarity with the MERS and the SARS virus, and this the severity of this disease is associated with the production of the cytokine storm. Till today, most of you know about what a cytokine storm is. Those who don't have the clear concept behind the cytokine storm, they can just ping me and I will release a video with that explanation also. And nowadays we have these such antiviral drugs out there in the market. Ivermectin is there, remdesivir is there to treat for the treatment, and it is helping us a lot. And today I have heard in the news also that there is this new variant which includes the Indian variant, and this variant is very pathogenic. It is spreading very fast, and this can give us very disastrous results because it is spreading very quickly as compared to the other two variants which we had back, especially in the Britain. We had the variant. Indian variant was till today was the most pathogenic form, but this new variant is a combination of both the earlier variants, and it is more deadly as compared to the existing variant. But don't worry, scientists will figure something out, and God is there with us every time. Okay, so our duty is to understand and to focus on our studies. Initially, I want to discuss a little bit about the coronavirus, how it looks like, how it is infecting the host cell, how it is multiplying. Then we will discuss how, in general, the viral genes can be a part of the host cell. That host can be an insect or a plant or the human. Then we will take that forward, how the viral genome is integrating into the human genome. That is the area of our interest. And is there any evidence before? Yes, it happened. We will discuss that. And since now the genome is there, the viral genome is there in the human genome, what consequences we will be getting, what we are facing. So we will discuss all these things one by one in this lecture. First of all, about the structure of coronavirus, which we most of us know that it is composed of the four portion. The first is the spike protein, then it has a membrane protein, the envelope protein, and a nucleocapsid protein. The spike protein is the one which helps for the integration, or you can say most appropriately in the easy word, for the entry into host cell. It binds to the receptor that is present on the surface of the host cell, and that receptor is getting triggered by another proteins present, and that's how it enters into the host cell. The nucleocapsid protein is kind of a protective environment, protective protein, which is protecting this single stranded RNA that is in this greenish color. So you have the two layers of the nucleocapsid protein, and it is encapsulating the viral mRNA. The membrane protein creates an you can say the envelope like a cell membrane around it, and the envelope protein is there for as a sort of, you can say, the envelopes. Okay, let's not discuss in detail about their structure because that is not the part of your course, that is beyond the scope of your MBBS. Okay, so now this is the viral entry. The first step is this is we are studying how it is infecting the host cell. Viral entered through the nasal or the larynx mucosa, right? And then it virus enters via the respiratory tract, peripheral blood, and reaches the host cell. On the host cell membrane, you have the ACE2 receptors, which are activated by this TMPRSS2. So TMPRSS2 is activating the ACE receptor. ACE receptor is very generous. It gives kind of an admit card to the virion, to the viral particles. Okay, you can come in. Virion will come and knock at the door. Thuk thuk thuk. The gatekeeper there at the door will ask for the admit card. They have the admit card which is given by the ACE2 receptors, and they will enter the host cell and will start infecting it. How they will are going to infect it? They have this viral replicase enzyme associated with it. Name is itself saying the replicase. So it starts making the replica of the mRNA of the virion. So viral mRNA is being replicated by this viral replicase enzyme that has an open ready flame, the similar things that we have studied in the molecular biology, right? And they will start replicating it. And few of the mRNAs have this tendency to make the proteins also. For all these process, they need some they need their food, right? To for the replication, they need few things. To survive, they need few things. So they survive on the host cell machinery. So they are, you can say, ditching the host cell. First, they are getting entry into the host cell. Then in the host cell, they are going to infect the host cell. But for that infection, they are going to eat food from the host cell itself by using their machineries. Okay, the virial proteins, with the help of mRNA and via integration via like the rough endoplasmic reticulum, they will travel to the Golgi apparatus. In the Golgi apparatus, the viral genomic RNA and everything will combine to make a very good, or you can say exactly similar replica of the virion, in which you have the M protein, then the nucleocapsid protein, and that single stranded mRNA in there. So exact replica of the virion which has entered into the cell, the host cell made two copies of it. And that entire process of making replicating the one virion into two is done inside the host cell using the host cell machinery. I call them as a kind of parasite. Okay, and now then they will be released, these particles will be released, new viruses will be released to infect the other host cells. So that's how they are infecting us. That is a too bad. And till now, our entire genomic sequence of the SARS-CoV-2 virus has been elucidated by the researchers, and it they share a lot of genomic similarity with the MERS and the SARS viruses. Okay, and this the new variant, the SARS-CoV-2, right? The SARS-CoV-2 is 30 kilobase pairs in the size, right? It has 13 to 15 open reading frames, which contains approximately 30,000 nucleotides. And its genome consists of approximately 38% of the GC content. GC content which has 11 protein coding regions, protein coding genes, with 12 expressed proteins. Right? The major culprit, if we talk about the virus structure is your spike protein, because spike protein is the one which is facilitating the entry into the host cell, thereby replicating the viral mRNA. Okay, this spike protein is made up of 1273 amino acid residues, and it has three subunits: S1, S2, S2 prime. So this is about how a SARS-CoV-2 virus looks like and how it is getting entry into the cell. Then the question comes, are there any mechanism by which the virus are able to integrate into the host cell genome? Yes, in virology, we study that the many viruses are able to insert their genetic material into their host chromosome. And there are three kind of integration. Actually, one is the mandatory integration. Second is occasional integration. And the third one is endogenous viral. That is the major focus for us. That is endogenous viral elements. Integration. Okay, mandatory integration is an for the few virus is an obligatory event during replication. Mandatory integration is an obligatory event during replication. In case of, let's say, the retrovirdae, pseudovirdae, metavirdae, myovirdae, they permanently integrate into the host genome. Okay, and they are usually seen with the prokaryotes, not with the mammalian cells. That is our major concern. That SARS-CoV-2 virus. Okay, the occasional integration is not necessary. Is not necessary during replication, but it does happen. Why it happens? Because uh, it sometimes, when after the integration, it facilitates the long term infection of the virus into the cell, and it also sometimes provide a advantage, because for us, because if we integrate such kind of genome into the host cell, then the toxins that are produced there can be used for the treatment of the various disease, right? So many bacterial toxins are carried out by the prophages. For this example, again, not an area of our discussion, but maybe for the ones who are working in the gene technology for gene therapy, or maybe to find out the antibiotics or something, they do study this in depth. Our major concern is the endogenous of viral elements, and these endogenous viral elements, they have integrated long back up, or you can say now they have fossilized into our genes, into the genes, or you can say more specifically, I will say the host genes, right? Right, through a rare, sometimes this process of integration can be rare, or you can say accidental, right? So based upon that, there are two kind of endogenous viruses. There are two kind of endogenous viruses. One is rare RNA virus integration. That is our case, the SARS-CoV-2 virus integration. And I will show you the paper how it is being integrated. And then is endogenous retrovirus. These endogenous retrovirus, if we talk about, they remain latent, okay, and they are passively replicated along with host genome, meaning that they are passed to their offspring, because when the entire genome is being replicated, I'm talking about replicated, not the transcribed, but it is replicated, then the daughter cells, the daughter chromosomes, daughter progeny will get this, but the host environment condition can however replicate the virus, and sometimes it leads to the viral transcription also, and which may produce the infective virus. So this infective virus, or you can say this is known as productive infection. So imagine you have the virion RNA that get inserted into the host genome. When the host genome is getting replicated, it becomes the part of the normal genome machinery of the host organism, whatever that host organism is. Now, when from the DNA, that is the genomic material, the RNA will be made, the transcription will be done, then that transcription part could make the activated virus, that could activate the virus, that is the productive infection. So imagine this was the mRNA. This was the mRNA of the virion, viral mRNA. And then this is the host DNA, right? And somehow it gets integrated into the host DNA. Now this host DNA undergoes replication. This will produce two copies, with the integrated viral DNA in between. When this will go under transcription, these green particles, these green inserted virion, they are going to make the mRNA. That is the viral mRNA. That can lead to the productive infection. And these green, the retrovirus genomes, which have become integrated into the germline, they are known as these ones. They are known as endogenous viruses, or in the short form ERV. Okay, so this is about the endogenous retroviruses, that how they become a part of the host genome, and then they can produce the infection when they are being transcribed into the host, inside the host cell. Now, our case is the rare RNA virus integration and the double stranded RNA. Now, our case is this rare RNA virus integration. That is our SARS-CoV2 virus integration. That is our major area of concern today. The double-stranded RNA, the single-stranded RNA, the single-stranded RNA negative. These viruses, they do not get transcribed. So they are unable to integrate themselves into the viral host genome, right? But still there are few viruses that are detected in the host genome, like this rhabdoviridae, DAE. This is found in kind of in insect genome. right? Then why we are discussing about the insect genome? For us, the major point to discuss is the LCMV virus, which is stands for lymphocytic meningitis virus. This is a known retrovirus, known retrovirus, LCMV virus, lymphocytic choriomeningitis virus, right? These viruses have been integrated into our human genome, and the studies have shown it and proved it to us. Okay, so now why we are actually discussing all this, the host genome integration, why the hell we are doing it? The question is, a person, a human become COVID infected. We did the RT-PCR test. He underwent all the medications, all the isolation period. After 15-20 days, the infection level goes down, measured by the symptoms, the visible symptoms, the coughing, the fever, and via the RT-PCR also. And the CT value is very well under the accepted values, and now we know that person is not COVID infected anymore. So he's not suffering from the COVID-19. He's not SARS-CoV2 positive, but still he doesn't have infection. He doesn't show any signs and symptoms of it. Even the cells doesn't show anything. But still, how come the RT-PCR is positive? Even after the months of the infection, people are showing the positive RT-PCR, though the cells does not show the infection in any of the other tests that are out there for to test for the SARS-CoV-2. That was the question, why RT-PCR is coming to be positive. So based upon this, the assumption or the debate has started between the researchers. Probably the viral genome has integrated into the human genome. But again, the debate took a hot like you can say the toll. Why? This is RNA, right? And human has DNA. The integrations we have discussed cannot be applied here. Why cannot be applied here? To get integrated into DNA, you need a DNA. RNA cannot integrate into DNA. So for that, RNA must be reverse transcribed via the reverse transcriptase enzyme to get converted into DNA, and that DNA can enter into the nucleus. RNA cannot enter the nucleus. It is there in the cytoplasm. To get entry into the nucleus, RNA has to become the DNA. And to become the DNA, RNA should have a friend enzyme known as a reverse transcriptase enzyme. But the problem is there is no reverse transcriptase enzyme with the SARS-CoV-2 virus. So he doesn't have friend. He has just the replicase enzyme, which is making the replicas of the mRNA. He doesn't have any friend which can reverse transcribe it into the DNA, and that DNA can enter into the nucleus and then be friends with the human DNA. See, I am DNA. And the human DNA will say, okay, that is a DNA, doesn't seem like to be mRNA. So they will be having friends, and then how it is integrated into the human genome. Now the question is, since the virus particles, the virus doesn't have the RT reverse transcriptase activity, who is doing that? Well, there is this line. We have these line 1 elements. What are these line 1? These are long interspersed nuclear elements. they are there in our human genome. Let's say it's too long, right? It's too long. So in between you have these line elements, and it has already proven in the past that these line one, they sort of act as the reverse transcriptase. They have the reverse transcriptase ability. So this line one has helped the viral mRNA to become the cDNA, and that cDNA enters into the nucleus of the host cell, thereby getting the place into the host cell machinery, that is us, become a part of us. So when after getting infection from the COVID positive patient and he becomes a COVID negative patient after several months of passing that infection, when the sampling is done, when the sampling is done, it has the cDNA, or you can say the DNA, the host cells are there. And these DNAs, when are run through the RT-PCR, they are going to show the positive signs, because the DNA, the DNA already has the integrated viral DNA complementary to it. That's how that answers the sort of a question that why even after several months of infection, the patients, or to say the recovered patients, are showing the positive RT-PCR. Okay, this is the paper that is a hot topic nowadays to discuss, that the reverse transcribed SARS-CoV-2 RNA can integrate into human genome of cultured human cells and can be expressed in patients' derived tissue. Not to go in detail with what they have done here, let's come to the results, what they have shown us via this diagram. So that was done actually via taking three approaches were used actually in this. One is this nanopore nanopore long read sequence they use. Then the Illumina pair and whole genomic sequence is used. Then the third one is used is TN5 integration method. Okay, not to go in that detail, also just for understanding how it is integration and how it is proved, I was just want to show you here. If you see, look clearly here, this is the pink one. Pink one is the SARS-CoV-2 entire genome, which is flanked by this human DNAs. So they make sort of an chimeric sequences that we have studied in the class also, what the chimeras are. And here, this is the point where they have been integrated. Target site duplication. Do you see this in the yellow color? These are the target site duplication, in which that's how they are becoming overlapped. This is the human DNA. This is the viral DNA, and they are becoming overlapped. They have some kind of a structural, you can say, the base pair similarities, right? That is provided us by the line, this line endonuclease recognition site sequence, which is indicated by these underlines here. So they help us for the integration of the viral DNA into the human genome DNA. These people, they have proven that in via the chromosome also, that at this chromosome junction 2.21.1, they have this nanopore alignment that is here, that is shaded by this yellowish line. So in nutshell, to summarize everything, yes, the viral the SARS-CoV-2 viral genome has been integrating into the human genome. And that is the reason the RT-PCRs are coming to be positive after even the several months or several days of the infection. Now, the question is it going to infect or produce an infection? Yes or not? Another question is, if the vaccines that are out there in market are going to affect us or not. These are two major questions that is boggling everybody mind. And the entire concept we are discussing is theoretical, right? And that is actually is going to be like a genuine case, because nobody has, no researcher has, scientists have figured this out to be true. Coming back to this, let's say they have, you have the vaccine, right? Vaccine has what? Vaccine has mRNA that gets integrated into the human genome, right? So your genome plus the human DNA. Oops, my bad, human DNA. You have this human DNA. And then in between, you have these virus particles, virus DNA to it. They multiply over the period of time. They are going to multiply over the period of time. Is it going to produce a COVID 19 itself? No, because to do that, what do you need? You need the entire virus particles to produce the infection, right? That is for sure. And to get the virus, what you have to do is that this DNA, the nucleic, the genetic material should be able to get transcribed into mRNAs, right? And that mRNA should be, first of all, the viral RNA, right? And that viral mRNA should have the viral proteins also, so that they get encapsulated in the Golgi apparatus, and then they will be able to produce the infection, right? This should happen, that we have studied in the beginning of this class also, that to produce the infection, the viral particle should be complete. It should have the mRNA, and the viral proteins should make the nucleocapsid protein, your M protein, and then other proteins, and then that makes the entire virion that can produce the infection. But actually this all does not happen. Why this not happen? This viral proteins are not be able to be produced fully, because when they will be produced, since they are not part of the human genome, the the immunity is going to kill those proteins, or engulf those proteins, or degrade those proteins. So you, the inside the host cell, there will be like few of the proteins, not all of the proteins. So whatever the viral particle, if it is going to make its own structure, this is not going to happen. Are you getting me? For the infective virus, you need its complete structure. And that complete structure is going to come from the genome that is there in the human. When the human genome is replicated into its mRNA, and the viral proteins will be made out of it, the host cell machinery, the human cell machinery will not be able to recognize it. For them, the system, for them, those proteins are invaders. They are going to maybe destroy it, degrade them, recycle them, whatever they will do. So this is just a theoretical aspect. So, or you can say, is they will be entire viral thing. If in a case it will produce the mRNA, that will be kind of a scrap that is not going to infect the system. So vaccine would be safe. Vaccine is not going to cause the productive infection inside the host cell. That takes care of the first one. Now, since let's say I am COVID infected, now my RT-PCR is positive. Are there any chances that maybe after 5-10 years, or maybe my kids are going to get this productive infection? The answer again is no, that is not going to happen, because the same reason, that if let's say my genome may produce the viral mRNA, and it may produce few of the viral M proteins, but still it won't be able, my body will not be allowed, my immunity will, the defense mechanism of my body will not allow to make the entire virus structure again, so that it can produce the infection. So the chances to build again this viral particle inside my body from my genomic sequence, the chances are very, 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 very negligible. So that is not going to happen, though the virus RNA is getting integrated into the human genomic sequence. I know this is a little bit weird, maybe complicated, But if you don't understand it, if you have any problems, any issues getting it, please, you can email me. For those who are unable to understand in English, I will, I know two languages, Hindi and English. I will explain in Hindi also. If you have any questions, you can just email me. You can write down my email, that is vgusp2013 @gmail.com. Okay, be safe and throw up any questions, any concern, maybe debate if you want to do. I'm ready for that. Good luck.